All right, so today's lecture is by Professor Francisco Raymo. He's a faculty in our department. He has been here longer than me in the department. He is a very well-known uh, chemist. Of late, well, last 10 years, he has been working in the area of photophysics and photochemistry. He uses quite a bit of these microscopes, so I thought he would be the ideal person to discuss about fluorescence microscopes, how the fluorescence is used in various biological systems. So that's what he's going to talk today. All right, Francisco.
Okay, hold on a second because I have, okay, I had all the faces in front of my slides, so I couldn't see my slides. Anyway,
So you can turn off all the people's pictures. And, yeah.
so I guess thanks Murthy for the introduction and for the opportunity to lecture within your class. So what I'll try to do today is to give you a general overview of fluorescence imaging. I give for granted that you all know what fluorescence is and how it works. And I will just try to show you how fluorescence can be used to acquire images using microscopes and how those images can be used to study biological samples. Okay. And I will be happy to give you copies of the slides later if you want. And of course, if you have questions, just stop me and ask. So I will start by showing you the structure of my favorite fluorophore, Bodibi. So we do quite a lot of work with this type of molecules in my lab. And what you have here on the right would be the absorption spectrum of the molecule in black. So this compound has a maximum at about 520 maybe. And then you can excite it at 480 nanometers. And of course, you all know that once you excite the molecule, it goes into an excited state, and then it goes back into the ground state by emitting fluorescence. And the trace in green will be the fluorescence spectrum. So you see the maximum is at about 540. So all this should be already familiar to you. What you may not know is that you can use that fluorescence to acquire an image. And what we are looking at here is a fluorescence image of this molecule entrapped into an hydrogel matrix. So the green color that you're seeing here is essentially the fluorescence that you see in the spectrum. So the microscope is detecting that fluorescence, and it is reconstructing an image on the basis of that. Now, optical microscopes have been around for a very long time, and they were already on the market in the mid-1800s. So this microscope here was commercially available already in 1872, and it was produced by a company called Carl Zeiss. And this particular one was designed by Ernst Habbe. You might have heard of Habbe. He's the main person behind the development of microscopy. And the way this works is that you put your sample here on the stage of the microscope, and then you have this tiny mirror at the bottom. So you might have a lamp, let's say, up here producing light. So the light bounces off the mirror, goes through the sample, and the light that is transmitted through the sample travels all the way up to the eye of the person that is looking through the microscope. So the samples are visualized on the basis of transmitted light. And again, this was already commercially available in 1872. Now, fluorescence was reported in the literature in 1845, so a little bit earlier. And this person, von Helmholtz, who was familiar with the initial reports on fluorescence by Herschel and Stokes, realized that by putting a fluorescence sample here, it should be possible to visualize the sample looking at the fluorescence from here instead of the transmitted light. So it suggested the development of a fluorescence microscope that could visualize samples on the basis of the emitted light instead of the transmitted light. And this happened in 1874. And it took a while for a fluorescence microscope to be developed. The first one showed up in 1911, And this is a diagrammatic representation of the very first fluorescence microscope. And it was developed by this person, Oscar Einstein, who was working in Austria. And he was actually working for a company. The name of the company is Karl Reichert. So this is not the name of a person, it's the name of a company. And as you can see, the actual microscope is very similar to what we had in the previous slide. But in addition, there is this big black box, which has a light source that is needed to excite the molecules that are in the sample and produce fluorescence. And the basic architecture of the fluorescence microscope today is actually very similar to what he came up with in 1911. This is a diagrammatic representation of a present-day microscope. And in modern microscopes, you also need to have a light source, just as in the case of Einstein microscope, except that today we use lasers most of the time. And then the light travels through a series of optical elements and then is bounced off a mirror. It goes through the objective lens. The sample would be here. So the molecules there would absorb the incoming light, and then the fluorescence would travel back through the objective lens all the way to the detector. And notice that the excitation source and the detector are on the same side of the sample, okay? And when detector and excitation source are on the same side, the term EPI is used. So this will be an epi illumination microscope. So I'm having here a few definitions. So epi illumination refers to the fact, let me go back, that excitation and detection are done on the same side of the sample. It is possible to move the excitation down here, for example, and have the two on opposite sides, in which case the term trans illumination is used, but epi illumination is much more common. And then the other important point and difference I would like to mention is that you can collect images in two different ways, in wide field mode or in laser scanning mode. And what I'm gonna do is to go back a second and maybe use this image as an example. Let's see if I can write on my screen, if I manage to, let's see. Does it work? No. Let's see. Then maybe. Yeah, okay. So this image, so the side of this image is about 30 migrants, okay? And you can collect these images in two different ways. The microscope can illuminate the entire area in one shot. So the whole area is illuminated, and then all the fluorescence that is coming from the illuminated area is detected at once, okay? In which case, we talk about wide field microscopy, okay? So all the fluorescence that is coming from the so-called field of view is captured at the same time. The other possibility would be, let me remove these lines if I manage. They don't go away. Why is it? And they don't go away. Okay. All right. So instead of illuminating the entire field of view, what you can do is you can point your laser into a small spot, let's say here. And then only the molecules that are here will produce fluorescence. So you first detect fluorescence there, and then you move your spot here and collect fluorescence, and then here and collect fluorescence, and then here and collect fluorescence. So you're scanning your laser, your excitation laser, across the sample and measuring fluorescence point by point. And of course, this is called laser scanning. Laser scanning, okay? And now let me do, where was the, here. Okay, now let me move forward. Okay, so laser scanning is used generally in combination with confocal optics. Now, this diagram over here is supposed to be similar to what we had before. So we have sample, objective lens, dichroic mirror, and laser. If we go back a second, it's the same here. Sample, objective lens, dichroic mirror, and excitation source. So this part is exactly the same. The addition in the other diagram, the additional element in the other diagram is one optical component here before the detector, which would be this black line over here, which has a tiny little hole in the middle, which is called pinhole. And the size of that hole can be controlled generally with the software of the instrument. And if you can squeeze that hole down, so you can make it really small, what happens is that the fluorescence that is above and below the focal plane is eliminated. So in other words, by making this hole really small, you can squeeze fluorescence in the vertical direction. So you can take a very thin slice of the sample. And what happens is that the actual volume that is observed, so in my drawing here, I don't know if it is still there. So I had like a circular spot, but in reality, the light is focused in 3 dimensions. So you have a little bit of light focused above and below the plane. So the shape of the observed volume is more like an egg, which is what we have here. So this one over here, okay. So this would be the plane where the light is focused, but above and below you also have fluorescence, and the volume that is observed as the shape of an ellipsoid, okay? And the distance between here and here can be controlled adjusting this pinhole. And generally, it cannot be, if you use something that emits in the visible region, it cannot be smaller than 500 nanometers, and the distance between here and here cannot be smaller than 200 nanometers. So the actual volume that you can observe at once, compared to the size of molecules, is very large. Okay, so you can have many molecules in this volume, and then this is scanned across the sample horizontally, as I was doing with my pen, to acquire fluorescence in the focal plane. So what you can do with a confocal microscope is to take sections of your sample. Let's say that you have something that looks like this, which is more like a ball, okay? So it is a 3-dimensional object. It's not a flat object. It will have a certain size in the vertical direction. So you can focus your light first in this focal plane, okay? And then you can scan your egg across this plane, point by point, and then collect the first image. Then you can slightly move the focal plane in the vertical direction. So for example, down here, and repeat the process and collect another image. Then you can move it down and collect another image. So you can take slices of the sample. And by comparing these images, you can even reconstruct, using the right software, a 3-dimensional representation of the sample. Later, I will show you one example from my lab, and I will show you how it is possible to acquire, by scanning the focal plane vertically, it's possible to acquire images that are at 90 degrees compared to the focal plane. I will show it to you in a moment.
Francisco, this the imaging is mainly for solid samples, or it is not for liquid samples?
No, this you can use also for liquid samples.
Okay.
So you can even do, like, let's say if this is a solution, like you have molecules that are swimming in a petri dish.
Okay.
You can still scan your egg, let's say, across the solution and acquire an image. Of course, diffusion will be significant. Some molecules will move everywhere.
Yeah.
So you will see probably fluorescence everywhere, but it can be done.
Okay. Okay. So, but it is generally used more for the biological systems or solid samples?
Since the samples are solid, but there is no reason why not to do it with liquids.
Okay.
Yeah, I mean, the microscope is compatible also with liquids.
Mm-hmm. Okay.
Okay. Then another important microscope is the one based on total internal reflection, which is very valuable for the detection of single molecules. So let me see if I can manage to explain it to you. In fact, actually, in this case, in this particular example, the sample would be a solution of fluorophore, so F stands for fluorophore, swimming in solution. So you have these fluorophores that are moving in solution. And then on top of the solution, you have a glass slide over here. And then the laser exciting the molecules is somewhere up here at an angle relative to the surface. And the angle has to be so large that the incoming light is totally internally reflected. So the light doesn't go through glass, it bounces off, okay? So it remains on the same side of the sample. But I'm not sure if you know what the evanescent field is, but let's say there is a little bit of light that is very close to the surface of the glass slide, but on this side, which is represented by this plot over here. So this is representing, it's upside down. So the intensity of the electromagnetic field against distance. So within the first 200 nanometers, there is some light in here, okay? Beyond that, there is no light. So which means that the fluorophores that are close to the surface can be excited, and the ones that are away from the surface cannot be excited. So using this trick, so total internal reflection, you can confine excitation only in this layer of the sample. And this is used to make sure that only a few of the molecules are excited and therefore emit at any given time. And this is very valuable for single molecule detection. And the way this is done is to use a prism, okay? So in this diagram, the exciting laser would be here. So light travels in this direction, then reaches the surface of the prism. It is refracted, it goes down, and then it is reflected. So this area, this shaded area here would correspond to the sample. And then the reflected light goes away. So these two arrows would correspond to this, okay? So by putting a prism on top of the sample and the objective on the other side, it is possible to take advantage of total internal reflection and only excite a few of the molecules at any given time. Then another important microscope would be the multiphoton microscope. And you probably have learned already that you can excite fluorescent molecules as a result of 1 photon absorption, 2 photon absorption, 3 photon absorption, and so on and so forth. Now, the molecules in each one of these 3 cases would then produce fluorescence. So the fluorescence that you detect is going to be very similar in all 3 cases. Might be differences in intensity, but in terms of position of the emission bands should be the same, more or less. But the absorption process is very different. In the case of 1 photon, you're going to use a fairly short wavelength. And then if you use 2 photon absorption, that wavelength is going to be half of the original value. And if you use 3 photon absorption, the wavelength will be 1/3, okay? So the wavelength requirements for 1 photon, 2 photon, and 3 photons are very different. Now, this is very valuable in the context of biological applications because biological samples are very sensitive to ultraviolet and even visible radiation. So if you can avoid using UV or visible light to excite the molecules, then you can also avoid damaging the sample. So if you can excite something with a much longer wavelength, in the near infrared, for example, then you can have avoid the photodegradation of the sample, and you can also achieve a much better penetration depth. And of course, 2 photon in most instances can be done with wavelengths longer than 680 nanometers. And of course, in the case of 3 photon, the wavelength could be even longer. The problem is that the probability of absorption goes down significantly as you move from 1 photon to 2 photon to 3 photons, okay? So in order to have the simultaneous absorption of 3 photons, you need to have a much higher power density. So you need to irradiate with much higher intensity. And in fact, the probability is proportional to the square of the intensity in the case of 2 photon, the cube in the case of 3 photon, and so on and so forth. But this correlation between intensity and probability of absorption can also be used to our advantage. And this is shown actually in this picture. In a second, I will explain with this diagram what these lines are. So here we have an objective lens on one side, an objective lens on the other side. So this one is used to induce one photon absorption. And as you can see, fluorescence is everywhere along the path of the light. This one is used to do 2 photons. And as you can see, fluorescence in this time is only in this tiny little dot, because only there is significant intensity for the molecule to be excited from ground state to excited state, okay? So it is possible to confine fluorescence into a much smaller spot by using 2 photons. And the same process is illustrated here. So up here, we have the objective lens that corresponds to 1 photon absorption, so this one. And down here it would be this one, okay? So in the 1 photon absorption, light, the focal plane of the lens would be here. So light is predominantly focused at this position, but there will be enough intensity in all this area to excite the molecules. So we're gonna see fluorescence here in the focal spot and also there. In the case of 2 photon, only at the focal point, the intensity is sufficiently high for this process to occur. So only here we are exciting the molecules effectively. So the fluorescence in this area and in this area, which is what we're seeing up here, is not detected. So we end up confining fluorescence into a tiny little spot. And then with the right instrumentation, what you can do is you can move the sample relative to the objective, which means that this spot can be scanned across the sample. Okay, so you can collect an image point by point, as I described earlier, but in 3 dimensions. So using 2 photon, or even better, 3 photon, you can acquire 3-dimensional images. Okay? Any questions so far? No? Right. Then another important technique is the so-called FLIM, fluorescence lifetime imaging microscopy, which is based on lifetime measurements. I assume that you know what the lifetime is already. And the way the microscope works in this time is to measure the intensity of the fluorescence over time. So let's say that we have, this is a cell, let's say that we have, that we are looking only at the fluorescence within this tiny square, okay? So the instrument will measure fluorescence in this square at time 1, then wait a little bit and measure it again at time 2, then wait a little bit and measure it at time 3, wait a little bit and measure it at another time, and so on and so forth. And then the instrument plots the measured intensity against time. So from the corresponding decay, the software extracts the fluorescence lifetime. So we can measure the lifetime in this specific spot only. Then we can move this square to the side, let's say here, and repeat the measurement, and then here and repeat the measurement, and then here and repeat the measurement. So we can map the fluorescence lifetime across the entire cell. And so in this color-coded scale, you see that you have lifetime values that are below 1.28 in red, and lifetime values that are above this number in blue. So the regions in blue have longer lifetimes than the regions in red, okay? So, and then you can extract information on the basis of this difference. You know? The fact that, for example, in the red regions, the fluorophore has a shorter lifetime means that it's probably interacting with some quencher, for example. So you can tell where fluorophores are by the information that the microscope is giving you in terms of lifetime. Okay? And in order to acquire these images, you need molecules that can generate fluorescence. And you need to be able to label biological samples with these fluorophores. Now, immunofluorescence is probably the most common way to label biological samples at the moment. And it all came out of the seminal contributions of Albert Kunz, who was a biochemist at Harvard University, who realized the potential of fluorescence in the context of studying biological samples. And so what they did is to take a synthetic fluorophore, fluorescein derivative, and attach it to an antibody. So this compound is called FITS, which is an abbreviation for fluorescein isothiocyanate. The isothiocyanate group up here reacts with the primary amino group of the antibody, and so the fluorophore becomes covalently attached to the antibody. So after this covalent conjugation, the antibody becomes fluorescent essentially. So you can see where the antibody goes in a given biological sample, measuring the fluorescence that comes out of fluorescein. And of course, the approach is not limited to fluorescein. Today, we have hundreds of different synthetic fluorophores that are specifically designed for this type of labeling strategies. You know, if you go through the Sigma Aldrich catalog, for example, you'll find plenty of examples with all kinds of fluorophores, fluoresceins, rhodamines, bodides, coumarins, that have reactive functional groups that can be used for this type of processes. Then, of course, antibodies have binding sites that can selectively recognize and associate with complementary targets. So you can, let's say, if you have a cell and you're interested in a certain subcellular component, you find an antibody that can bind that particular target. You attach your fluorophore and then you incubate the cell with the tagged antibody. The antibody will find its way to the target, and then you can visualize the target by measuring the fluorescence that comes out of the synthetic fluorophore that is connected to the antibody. Now, another class of extremely valuable fluorescent probes are the fluorescent proteins. And as you might know, the Nobel Prize in Chemistry in 2008 was given to Chalfi, Shimomura, and Tsien for the discovery and development of the green fluorescent protein, which was the very first of these fluorescent proteins. Today we have, I don't know many, but plenty of fluorescent proteins with different colors. Now the green fluorescent protein, which is generally abbreviated to GFP, was originally isolated from these jellyfish, and it can be encoded genetically in cells. In other words, you can design your cells or you can instruct your cells to synthesize on their own the green fluorescent protein. And then you can measure the fluorescence that comes out of the green fluorescent protein to image the cells. And also, you can instruct the cells to synthesize the GFP in specific locations so that you can send the fluorescent protein onto the target that you are interested in imaging. And this can also be done with fully developed animals. If you search online for glowing animals, you'll see all kind of strange pictures with rabbits and pigs and all kind of animals that do express GFP. And as I said, in addition to GFP, today we have plenty of other fluorescent proteins that produce yellow, red, and magenta, and all kind of colors. And the combination of fluorescent proteins and synthetic dyes, as well as antibodies, is extremely valuable to image simultaneously different targets. What we are looking at here is a collection of images of cells. In each case, you see many colors. Each color corresponds to a different fluorescent probe, which can be a synthetic dye connected to an antibody, a fluorescent protein, and they can be imaged at the same time. For example, here we are looking at green, orange, and whatever that color is, purple maybe. So these are three different fluorophores sent to three different targets. And by imaging them at the same time, we can see how these targets are positioned relative to each other in the sample. And as I will show you later, not only it is possible to image structures and gain information on the structural features of cells, but it is also possible to monitor dynamic events. So you can acquire images sequentially. Essentially, you can record the movie and you can watch the different targets move relative to each other. I will show you one example in a moment. Now, one limitation of this technique is that the resolution of these images is limited to the micrometer level. And of course, molecules are much smaller than that. So it is not possible using conventional fluorescence imaging to investigate structural features at the molecular level. And this is because of the phenomenon of diffraction. So if we go back for a second to the diagram that I had earlier on, I told you that the sample is over here, and the fluorescence that is generated at this point has to go through the objective lens, which is then focusing the fluorescence on the focal plane of the lens, and there the detector is measuring the intensity. So if we, let's say, zoom in this part, what we would see is the fluorescent molecules on a glass slide, and then the emission is collected by the objective lens, and as I said, projected on the focal plane of the lens. Now, it is not possible to focus light with a lens without diffracting it also. Diffraction goes along with focusing. And the problem is that on the focal plane, the fluorescence will show up as a diffraction pattern. So even if you have just one molecule here, a tiny little molecule, it will look like a large pattern on the focal plane. And the dimensions of the pattern are mostly related to the wavelength of the light that is emitted. And in the case of biological applications, fluorophores that produce fluorescence in the visible region are used. So the wavelength is at least 400 nanometers. And the radius of this inner disk, so the distance from here to here, is approximately half of that wavelength. So if the wavelength is, let's say, 400 nanometers, the distance between here and here is going to be 200 nanometers, and the entire pattern is going to be much more than that. So if you have a tiny little fluorophore that is, let's say, 1 nanometer in size, it will show up as a huge pattern that is hundreds of nanometers in dimensions. Okay? So there is a significant mismatch between the real size of the fluorophore and its image on the focal plane. So if you have two fluorophores in the sample, one here and one there, the microscope will not see two small dots. It will see two very large patterns. So if the fluorophores are very far away from each other, and for very far, I mean several hundreds of nanometers away from each other, then the patterns can be resolved and the fluorophores can be distinguished. But if the fluorophores are close to each other, right, it is not possible to distinguish one from the other because the microscope will see two overlapping patterns. And this is why the resolution of the images that I showed earlier is limited to the microscale. Now, a solution to this problem came from the work of Betzig, Hell, and Merner, who were awarded the Nobel Prize in Chemistry in 2014 for the development of so-called super-resolved fluorescence microscopy. Now, Merner was the first person to show that the fluorescence of just one molecule can be detected under the right conditions, but still it can be detected. And then Betzig designed a strategy based on single molecule detection to overcome diffraction that is also taking advantage of the ability of certain fluorophores to switch their fluorescence. I will show you how that works in a second. And then Hell developed a method that is instead based on stimulated emission, which I'm going to explain now. Okay, so let me. How are we doing on time? We still have 10 minutes, I guess.
So 15 minutes.
15? Okay. So I told you earlier that the microscope is focusing, the objective lens is focusing the light in the form of a diffraction pattern, which has an inner disk and then a series of rings around. Okay. Now, most of the light is actually in this disk. So to simplify, we can get rid of the rings. And the dimensions of that disk are going to be, let's say, maybe more than 400 nanometers. Okay. So what he realized is that you can generate a second spot much larger which, however, has a hole in the middle. So light is everywhere, but not in that tiny hole. And then he realized that those two spots can be overlapped on top of each other, which is what we see here. Okay, so the dotted line here would correspond to the size of this spot, and then this would be that, and the hole in the middle is shaded in there. Okay, so it then did this with two different lasers with two different wavelengths. The wavelength of this beam is exciting the sample, so it's exciting the molecules from S0 to S1, let's say somewhere over there, and then the wavelength of that is, according to its definitions, depleting the sample. So the wavelength of that is encouraging stimulated emission. So it's pushing the molecules from S1 back to S0. And by doing so, that beam prevents spontaneous emission. So in this area, but not in the hole in the middle, the molecules cannot emit. So by doing so, the area that is effectively producing spontaneous emission is reduced to this part only. So this depleting beam is eliminating emission from here. So it's making this part much smaller. And then by scanning these two overlapping beams across the sample, like in a conventional confocal laser scanning microscope, one can acquire a fluorescence image that has a much better resolution because the size of the fluorescence spot is much smaller. Okay. Now I'm going to show you. Where is it? Here. I will show you what Bedzik did instead. So as I said, he relied first on the discovery of Werner that the fluorescence of single molecules can be detected as long as the molecules are sufficiently bright. And then he realized that if the molecules can also be designed to be switchable, then they can be distinguished at different intervals of time. And he actually did this not with synthetic dyes, but with fluorescent proteins. So the idea would be to have molecules that can be switched from fluorescence off to fluorescence on. So let's say that you have two of these molecules right next to each other, but only one is on, right? Then the microscope can detect the diffraction pattern of the molecule that is on only. And one can detect that particular molecule at the single molecule level if the detector is sufficiently sensitive. And by playing some mathematical tricks, one can find the coordinates, so X and Y, of the center of the pattern. Now, those coordinates are going to be the coordinates of the position of the fluorophore. And then after having localized that particular fluorophore, one can switch it off and turn the other one on and repeat the process and find the coordinates of the second one. And by doing this over and over, one fluorophore at the time, it is possible to localize the many fluorophores that are in the sample, even the ones that are close to each other, as long as they can be switched on and off at different times. So basically, he realized that closely spaced fluorophores can be separated in time. Okay. Now the limitation, though, is that you are collecting information over a very long time because you need to map the position of thousands of fluorophores sequentially. Right? In a conventional image, you image all the fluorophores at once. Here you have to do them one by one, essentially. And it takes time to go from one fluorophore to the next, and then to the next, and then to the next. So acquiring one of these images might require a few minutes. And during the time that is required to localize the fluorophores cannot move. So there cannot be any significant diffusion. Otherwise, the whole idea falls apart. So there are some limitations, but still, the final result is the ability to acquire images with a resolution that is of the order of a few nanometers. I'm going to show you one example from my lab. So this molecule is switching back and forth within two states. So this is not fluorescent, and this is fluorescent. So these dots that you see here, that kind of turn on and off, are individual molecules that are switching between these two states, okay? And this experiment was done by trapping the molecule in a polymer film so that the molecules cannot move. Let me play this again. So the molecules cannot move because, as I said, diffusion has to be restricted. So the dots that you see in certain positions are molecules fixed in place that are switching back and forth within these two states. And one can adjust the irradiation intensity for the photochemical conversion of this species into that species, as well as for the excitation of the product, in such a way that the probability of finding two of those dots on next to each other is 0, so that they can be distinguished in time. So if I take all the images that we had in this movie and put them together, you see that the sample has many molecules, but we are not switching all of them on at the same time. We're switching them on one by one so that they can be separated in time, even when they are very close in space, okay? And this can be used to, again, overcome diffraction and acquire much better resolution. So for example, we can attach this molecule to an antibody and then image the tubulin structure of a given cell. And this would be a conventional diffraction limited image. It's kind of difficult to see the nanostructures of the tubulin filaments, but by switching the molecules, you can clearly see these nanoscale filaments, okay? So we can improve the resolution and go down to the nanoscale by taking advantage of switchable molecules.
Francisco,
yeah.
The question I have is, so when you say switching, you had as off and on. So you had to irradiate that with one light, and then you had to irradiate the product with another light to induce the fluorescence. Yeah, here.
It's done with two different lasers.
So you had to have two lasers.
One laser has a wavelength of 355, and that it uses the photochemical conversion of this species into that. And then we have a second laser of 535, which excites the photochemical product to produce fluorescence.
But do they have to be synchronized or something, or no?
So this is done with CW lasers. So they're both on all the time.
Oh, it's on all the time.
Yeah, but you need to fine tune the intensity of the two beams, so that, because you don't want too many molecules to be in this form at the same time. So the intensity of the UV beam is very low, so that only a tiny number of molecules is on at the same time. And the intensity of the other one is very high, because you need to make sure that you're exciting them and detecting them at the single molecule level.
So once it goes to that excited state, it emits, and then it goes back to the original compound in milliseconds?
So it emits, it goes back to the ground state in this form, and then this thermally switches back in milliseconds.
Okay. So that's how it disappears by itself.
Yes.
Okay.
Now, in milliseconds in a polymer film, which is fairly rigid, in solution is faster.
Okay.
Professor Raymo, followed up with this question. These 500 nanometer photons won't convert the opened one to closed one?
I didn't understand. Say it again.
And the fluorescent molecule, the ring opened structure, may not go back to the closed one with the visible laser?
Oh, no, because so this is the photochemical step that is responsible for going from here to here is the cleavage of this carbon-oxygen bond. Okay.
Okay.
And once this is chopped, I mean, visible light is not really doing anything to this.
Okay.
This is not like, in the case of, let's say, the spiropyrans, you can use visible light.
Yeah. Oh, okay.
But here,
yeah, that's what I was. Yeah. Okay.
Right. So, and then I had a few slides to discuss dynamics. I'm not sure if there is enough time though.
Yeah, you can finish it, I think. Yeah.
Okay. So it is possible to use fluorescence microscopy also to study dynamic processes. The most common method is called fluorescence recovery after photo bleaching, or simply FRAP, F-R-A-P. You may have heard of FRAP. The way it works is that you use a conventional fluorophore, you label your sample, and then you irradiate a small spot with very high intensity so that the molecules that are in that spot only bleach. In other words, they decompose irreversibly. And they are essentially removed from the sample. They are still there, but they're just not emissive, okay? And then after this bleaching step, you can acquire a conventional image. Fluorescence will show up everywhere apart from the area that was originally bleached. So we're turning fluorescence off in this spot only, okay? But if these molecules can diffuse in the sample, if you wait a certain amount of time and then acquire an image, by that time, let's say this molecule might have moved back into here, and this might have moved back into here. So the fluorescence in this area is recovering. That's why this is called fluorescence recovery after photo bleaching. So an image after a certain delay will again show fluorescence in the initial area. So by measuring the correlation between fluorescence intensity in here over time, so the fluorescence will go up, one can extract diffusion coefficients, okay? One limitation of this approach is that the bleaching step requires very harsh illumination conditions. You need to illuminate with very high power densities in the presence of oxygen for a fairly long time. So for some biological applications, this is a problem. A better approach would be to use photoactivatable fluorophores, which are just like in the case of the molecules that Betzik used and the molecule that I showed. These compounds switch from one state that is not emissive to a state that is emissive. And again, as a result of a photochemical transformation, you can, if the design of the molecule is right, you can do it with very low illumination intensities. So you can selectively switch fluorescence on in this spot only by illuminating the sample at the right wavelength. Then if you acquire an image, fluorescence will show up here only. And then if the molecules can diffuse, the fluorophores will go out of that area and end up in other regions. So in this case, the fluorescence here will decrease over time. But again, from the correlation of fluorescence intensity and time, you can extract diffusion coefficients. Now, the advantage of this approach is, again, that you can use mild illumination conditions for the activation step, while in the case of bleaching, you need to basically blast the sample with light. And also you're turning fluorescence on against a dark background. So it's easier to detect the fluorophores in this case. But the problem is that at the beginning, there is no fluorescence. So it's difficult to decide where to do the activation because you cannot actually see what the sample looks like. So another approach, which is what we are doing, is to use molecules that are fluorescent from the beginning, but then they change the color of their fluorescence. So let's say that you have a molecule that is in an initial emissive state, which I'm calling state A. So you can acquire a conventional image and see how the sample looks like. Then you do your activation. So only in this spot, the molecules are switched to a new state, which produces fluorescence in a different spectral window. So you can then acquire an image which would show fluorescence here only. And then over time, these molecules would diffuse in other regions, and you would be able to see where they go by collecting images over time. And from the correlation of emission intensity and time, you can extract diffusion coefficients. So I'm going to show you one example, and then that's it. So this is one molecule that we made a while back, and it switches from this state to that state. And as the molecule undergoes this photochemical transformation, the fluorescence shifts batochromically. So this guy produces green fluorescence and this one red fluorescence. And so we can image the two in two different channels of the microscope that can detect fluorescence in two different spectral windows. Say it again?
No, I think he's leaving, that's all.
Oh, okay. I'm almost done. So here, the photochemical reaction is happening in this square only. So only in here, green is converted into red, and you can see that the green color disappears and the red shows up. So we can take advantage of these changes in color to monitor diffusion. So here we are looking at an embryo. It's a Drosophila embryo. It's a live embryo injected with the molecule in the initial state. And we are collecting two images at the same time in two different detection channels. So here we are looking at the spectral range where the green species produces fluorescence, and here in the spectral range where the red species produces fluorescence. But at the beginning, there is no red species. But we have the green fluorescence, so we can actually see where the cells are. These rings that you see in there are the cells that are in the embryo. So if you use a conventional photoactivatable fluorophore that is completely off at the beginning, you would see something like that. You cannot tell where the cells are. You cannot decide where to do the activation. But here we can clearly see where the cells are because we have fluorescence from the start. Then you can point your laser here and you can induce the photochemical transformation. So the color changes from green to red. So we generate the new fluorophore in the same spot, and then we can record the movie and watch the fluorophore move within the embryo. So we can monitor a diffusion process over time. And this is done with the confocal microscope. So if you remember, I told you that the microscope can measure fluorescence in a plane. So if we look at the changes of the fluorescence in this spot, in that particular plane, we see that over several minutes, the spot becomes wider and wider. And from these changes, you can extract diffusion coefficients. And also, you can then, as I described many slides ago, move the focal plane, let's say a little bit up and repeat the process, and then up and repeat the process, and then up and repeat the process. And by comparing these different sections of the sample, you can then establish if there is diffusion in the vertical direction. You can, in other words, reconstruct an image that is at 90 degrees relative to this plane, which is shown here. And then in here, we see that the molecules are now moving vertically. The fluorescence is only moving horizontally. Okay, so this is just to show you how the confocal microscope can be used to gain information in 3 dimensions and not just in a single plane. And I think this was my last slide. Yeah. I guess I'm gonna stop.
Okay. So let's see whether the students, anybody has got any questions. Okay, as you would expect, no questions. But it was good. It was
Thanks.
very good